So we've got this pace of speciation, when it occurs due to allopatric or parapatric or sympatric speciation. Now we're going to talk about reproductive barriers, pre- and post-zygotic isolation. Pre-zygotic isolation, what does pre mean? Before. And zygote, do you know when that forms? Because you've taken, family life. When does the zygote form? Fertilization. You got a sperm fertilizing an egg, and then you get a zygote. So all this happens before the sperm is able to fertilize the zygote. Pre-zygotic isolation can take lots of different forms. Here we have the sea slug. Sea slugs mate due to a pheromone that they put out into the water. Literally, these sea slugs get together, they put out this chemical, and the chemical drives them into a sexual frenzy. They all start mating with each other. It's crazy. Unless there's a mutation, and a sea slug doesn't have the receptor for the pheromone. At which point, that poor sea slug's looking around like, why is everybody mating? I'm just not in the mood. It's unfortunate. Crickets. Crickets chirp to get attention to mate. They're like, listen to me, I sound really good. This species of cricket oftentimes is defined by their song. If a cricket cannot sing that song because its hairs are a little bit in the wrong place, you've got the wrong pitch, all of the other crickets ignore it, and it's left out of the mating dance. These are pre-zygotic isolating mechanisms. They happen before mating can occur. They stop mating from happening. This happens prior to insemination. Birds dancing is a pre-zygotic mechanism. If you dance wrong, you're not mating. It's sad. This is as attractive as it gets with birds. And if they get that mating dance wrong, it's over. No reproduction. There are 5 mechanisms. You might have habitat isolation, temporal isolation, behavioral isolation, mechanical isolation, and gametic isolation. We'll go through each. First up, we've got habitat isolation. This makes sense. Listen. A geographic barrier makes interbreeding between populations impossible. All right, so there you are. You guys have, I'm not going to talk about your experience mating. Hold on, let's go a different route. But you understand the concept. If there's something stopping you from getting to somebody you want to mate with, can you mate with them? No. If there's a mountain between you, you don't want to walk over it because you're lazy. Does that make sense? There's the whole, if I could walk 500 miles. You know that song? Yeah. Contenders? Oh, yeah. Who's going to walk 500 miles? I mean, really. There's probably somebody closer. If long distance relationships, right? Imagine having to, I don't know, you live in Virginia and the person you want to mate with lives in England. There's a mechanism there to stop you from mating. It's called the ocean. That is an allopathic mechanism that prevents mating. This is pre-zygotic because it stops you from being able to mate. Another example, habitat isolation. Thinking about with parapatry, if you mate on your food and you prefer to mate on bananas versus mating on mangoes, you're going to be isolated from the other organisms even before you have the opportunity to fertilize an egg. We've done experiments with this in the lab because scientists don't have other things to do. What they did is they took a population of fruit flies. Fruit flies have a very rapid reproductive cycle. It's about 14 days, start to finish, having a new generation. 10 days, actually. So they took one population of fruit flies that were all the same. They bred with each other, life was good. And then they said, all right, we're going to put half of you guys, randomly selected, into this container with just maltose food, sugary food. And another half into this one with starch as their food. And they let them sit that way for about 14 generations. After the 14th generation, they put them back together, and they found that The ones that ate maltose food only would ever mate with the ones that ate maltose food. They were completely different species in 14 generations because you isolated their habitat. If you were to take a group of beetles, split that group of beetles, you'd start seeing isolation. This is, again, allopatry. They aren't able to mate because they can't cross that river. You could have temporal isolation. All right, so we had geographic isolation, now we have temporal isolation. What does temporal mean? In relation to time. Due to the timing of the mating cycle, interbreeding between two populations may not occur. You have to time things right. You have to be not just in the right place, but you have to be in the right place at the right time. It would be very difficult for one of you to mate with, I don't know, Abraham Lincoln or Martha Washington because they're dead. You are not in the right place at the right time. Frogs, for instance. There are two groups of frogs. You've got spring frogs and fall frogs. Spring frogs mate in the spring. That's why they're called spring frogs. Fall frogs mate in the fall. Therefore, this group of spring frogs will not mate with the group of fall frogs. Because in the fall, when the fall frogs are mating, the spring frogs are trying to eat and get ready for winter. They're not interested. They don't care. Fall frogs are like, last bit of summer, let's do this. Cicadas. Oh, welcome to Virginia, land of the cicadas. It's been a few years since we had a mass emergence. It's going to happen again. These things will be everywhere. But in the summer, doesn't it sound? It sounds like science fiction lasers going off all over the place. That really loud buzzing sound that you are so used to, it takes you a second to stop and listen and be like, oh yeah, I know the buzzing sounds you're talking about. It happens all the time, every day. Those cicadas are a great example of temporal isolation because you have a group that emerges every year. So yeah, they come out and they're like, yay, we're going to mate. You have a group that emerges every 3 years, one that comes out every 5 years, and one that comes out every 17 years. Literally, they live their whole life, 17 years. They live underground without seeing the sun. And they're like, let's crawl out for a day and mate. They come out, they shed their shells, they live for a few days. They have to live on what they've stored inside of them because they have no mouth. You will never be bitten by a cicada. Yeah, their whole job at this point is mate. Use up that energy. Mate as much as possible. Lay some eggs. Die. If you are a 17-year cicada, it's kind of going to be, you're not going to run into the 5-year cicadas. You're just not going to be able to mate. There's temporal isolation there. You're in the wrong place at the wrong time. Field crickets, again, you got summer crickets, fall crickets. They have different songs that separates them, but they also mate at different times. So, temporal isolation, be in the right place at the right time. Behavioral isolation. Different populations signal their mate in different ways. And if you misread those cues, you lose. One example, probably the best known example, is bird song. You've heard bird song before, right? Birds singing. And you think, oh, the birds sound really nice today. Birds are saying one of two things. Get the hell out of my area. Please mate with me. Those are the two things birds are saying all day when they're singing. There is no, like, let's just talk about the day. It's, go away, I'm a bird, this is my place, or, hey, I'm open for business. Come out over here, let's mate. The sad part is, the species is determined by the song, right? So if you have one member of that species has a mutation and sings the song wrong, either the bird's out there singing and saying, stay off my lawn, and then the other birds hear, I'm ready to mate, and that'd be really annoying. Or the other way, they're like, I'm ready to mate, and what the other birds hear is, you're attacking me. I want you dead. Behavioral isolation. Insect mating dances. I did a lot of studies on fruit flies. Fruit flies have a very, very particular stance they go through. They have to go through the exact same ritual for each species, and it differs based on the species. You can see my fly mating dance. First off, it's all about the orientation, right? This chair is my female fly, right? So what ends up happening is the male flies in from the side, because eyes have, there's going to be way more about insect population than you ever want to know, but welcome to biology. Flies have eyes on the side of their head, so he comes in, he's like, I'm landing over here, right where you can see me, because I'm not threatening. He's like, check me out, I'm going around you, I'm checking you out. I'm going the other way. Does that 2 or 3 times. That's called orientation. He just like, I'm here, please don't leave. Then he starts to vibrate his wings. This is where it gets tricky, because they vibrate their wings at a very specific frequency, and it causes a song to happen. We can't hear it, but the females of their species can. If they get that right, they can move on to the next step, which is called following. Typically, the female, if she doesn't like a song, will just fly away. But if she likes the song, she'll walk away. You know, mixed signals, right? Well, she's not flying, she's just walking, so she probably wants me to follow. So the male will follow the female. After that, the male licks her genitalia, but that, you know, that's how flies roll. That leads to copulation, and that copulation takes about 2 minutes, at which point both are just locked together. And then there's rejection, where the female's like, and now I'm done with you. And that's what, it has to follow those steps. If a male tries to copulate before licking, it doesn't happen. It has to follow a certain set. If it doesn't, the behavioral isolation kicks in, and this ends. Fun story. So part of my graduate research, when I told you about dissecting flies, was I had to watch them mate. Literally, hours of put them into a tank, watch them mate. When they mated, I had to pull them out of the tank. So as soon as I had a mating event, I took, you guys know what a pipette is, right? I had a pipette attached to a tube, and I would suck them out, like, literally, with my mouth, into the pipette, and then blow them into a small container of food. The most important lesson I learned from that was always put cotton into the pipette, because I swallowed so many of these things. They're like, and we're mating, and oh god, now we're in his stomach. Humans have behavioral dances we go through, and it's based on our cultures. Eye contact can be many, many different things. Eye contact could be a signal for the advancement of sexuality, but it could be a signal of dominance. You could look at somebody and be like, all right, I'm staring. You've heard of that, staring somebody down, right? I had to teach a class the other day for my wife on SDV class. You guys remember that? Mm-hmm. I screwed that up. I had to teach the part about good communication skills, and apparently, so we're teaching about maintain eye contact. You know, you nod, and you make eye contact's important, and it's a way of communication. So eye contact has lots. I didn't screw it up that bad. I did say, I misunderstood the point. So she's like, active listening. I was like, yeah. So you look at them, you nod your head, and you ask a question now and then. You know, even if you're not paying attention, you do that. She's like, no, you pay attention. And I was like, but they won't know you're not paying attention. That's how we get through our whole lives. I mean, heck, half of you guys are doing that now. But anyways, humans have isolating mechanisms. Eye contact is one. If you misread the cues, you will not be having sex. Does that make sense? Mechanical isolation. Morphological features of the organism prevent the two from breeding. The whole point of this is to stop the egg from fertilizing, the sperm from fertilizing the egg. For instance, here we have 2 dogs. They may be able to, you know, if we got the sperm and the egg together, there could be reproduction. But for these 2 dogs to mate, it would be very difficult. The much larger female, for instance, would not be able to mate very well with that very small male unless the small male had a step stool. That makes sense? There's something mechanical stopping that. Here's another example, copepods. The copepod's genitalia is covered by plates. If they were to go to mate and their plates don't match up, literally you can't get the genitalia to come together. You don't have copulation. It's impossible for it to occur without like a can opener. So those are your isolating mechanisms. There's one more. Gametic isolation. All right, here it is. Everything is worked out. You got the right dance. You got the right song. You were in the right. It was the right time. You were in the right place. All the parts lined up. You have successfully mated. The sperm is heading toward the egg. They run into each other and bounce off. With gametic isolation, the sperm cannot fertilize the egg. There's a chemical problem preventing the sperm from fertilizing the egg. This happens a lot in the ocean. I don't know how much you know about oceanic life, but pretty much there are times you guys don't want to know what's the top layer, like the top thin part of the ocean. You know, the part that you suck in when you fall under a wave. A lot of that is fertilized offspring, fertilized eggs and sperm. Some larvae in there along the top. There's a lot of stuff in there. Sea urchins. In order to get a sea, I used to run this experiment. It was great. In order to get a sea urchin to release its gametes, and they release them as a cloud, you inject it with saline. If you inject it with saline, it's not how it happens in the ocean. In the ocean, it's time based on salinity. But if I was to inject a sea urchin with saline, they will just put out a cloud of gametes. Big cloud of, like, sperm and eggs. And you could collect it. So what we did was we collected it, and then we took a container of sperm and a container of eggs, and you put them onto a microscope slide and watch fertilization happen. It was way cooler than the stupid paper lab you guys did. The issue is if you have a whole bunch of sea urchins all at one time releasing their sperm and eggs of different species, how do you guarantee that they don't run into each other and fertilize the wrong species? Well, it comes down to chemical signals on the egg. So sperms only can recognize an egg of their species. That's gametic isolation. So the sperm and the egg. Those are all the prezygotic mechanisms. Don't worry, there are far fewer postzygotic mechanisms. In fact, there are 2. As far as postzygotic sexual isolation goes, this occurs after the egg has been fertilized. Everything happened with prezygotic, now we're at postzygotic. We're after the egg is fertilized. J.D. Haldane said that when in the F1 offspring of 2 different animal races, one sex is absent, rare or sterile, that sex is the heterogametic sex. Hetero means different. The heterogametic sex in humans is the male, because we're XY. In butterflies, it's the female, because she's WZ. So, for humans, for a lot of mammals, it's that XY gene. If you're XY, you're most likely to be sterile first. The female is not. Pretty much what that means is the large X effect. Males will become sterile before females. Male sterility is one form of postzygotic isolation because you're stopping the male from being able to breed. Another form is called hybrid inviability. Inviability means it cannot live. So yes, you have a sperm and egg fertilized, but shortly after that fertilization event, the embryo stops developing. The organism is never born. The organism never develops to a point of adulthood. And as we mentioned, the male becomes sterile first, hybrid sterility. If an interspecies spouse is viable, that is, it can survive to adulthood, it may be incapable of breeding with other organisms. Probably because of polyploidy, where after meiosis in that offspring, the chromosomes don't line up, so it doesn't produce eggs and sperm that are viable, that can live with the other organisms. So you have hybrid sterility and hybrid inviability. And it's pretty much going to be determined by what's known as the large X effect, that males, the heterogametic sex, will become sterile first. That's where the issue is going to be. It turns out prezygotic isolation happens way faster than postzygotic isolation. Two species are going to act like two different species before they are physically incapable of mating. And that's where we get so many of our issues with the biological species concept. These species are acting totally different, and over time are becoming different. But they don't have the biological or the chemical mechanisms in place to prevent fertilization. It's just behaviors change faster than biology. When two species that have been separated come back together, there will very likely be prezygotic isolating mechanisms in play. They may not be completely separated as far as postzygotic goes. That is, they may not want to breed together, but they can. But over time, if you keep those species apart and they keep breeding with just the ones they want to, you're going to end up with two distinct species. So prezygotic barriers are going to reinforce species boundaries. When you have a ring species, it's going to be prezygotic barriers in that hybrid zone that stop two from mating as much as possible, stopping this from becoming one large population. As you get reproduction continuing, if the hybrid is less fit than the parents, that's going to also reinforce speciation, because it means the hybrids don't get to mate as much. So you don't see mixing of genes.